Good afternoon, everyone. Welcome to your subject in Nat Site 22. So, in continuation of our topics last time about microscope and the study of microbial structure, microscopy, and specimen preparation. So, last meeting, we did discuss about how is microscopy done, no? Starting from the discovery of microorganisms and um, about the um, explanation behind the lenses and light bending for each type of microscope. So as you can see on the previous slides, we have the different resolutions, the magnifications, the working distance, and the different types of microscope like the dark field and the phase contrast and interface differential interference contrast and the fluorescence microscope and how to prepare and staining of specimens. So whereas we use dye, especially for bacteria and yeast to identify them whether they are for whether they are gram positive or negative for gram staining and whether they are mycobacterium under acid fast staining. So in continuation to the previous topics on microscopy, we're going to discuss now the electron microscopy where we stopped on the previous video. So please check the video, the link that I have sent you on the Google Classroom. So in continuation to the electron microscopy, we have this is just the beams of electrons that are used to produce images. And if you remember last time, microscopes are the most high-tech microscope we have, no, so far. And in the Philippines, we even only have one here. And as shown in the previous photo, is just as big as a classroom. So because they use electrons, which are used to produce images. And we have wavelength of electron beam is much shorter than the light. So the electron beam is shorter than the light, resulting in much higher resolution. So it's more efficient, most effective way of and higher spec, no? So as you can see in the figure, this is the range of microscope. The range of the electron microscope is higher. And the scanning tunneling microscope. No? As you can see, the different cells that can be seen here in using the electron microscope are atoms, level, amino acids, proteins, viruses, no, in which a natural light microscope can only identify up to the mycoplasma. So now we it's briefly discussed here. Just So it's briefly discussed in the transmission electron microscope that electrons scatter when they pass through thin sections of a specimen. So if you, when light is transmitted, so light actually um, reflect back or they are scattered, no, when they pass through thin sections. And transmitted electrons, therefore, those that do not scatter, are used to produce the image. And denser region in the specimen scatter more electrons and appear darker. So that's how the electron microscope is transmitted. And this is shown here in this figure. The transmission of electron microscope. So we're in, this is uh, the which with using tungsten, no, filament, the electron gun, and these are the condenser, the specimen, and the object lens. and the projector lens magnet, and the final image of fluorescent screen or on photographic film is lifted aside in this portion. So for the specimen preparation on electron microscopy is different from the um, previous explanation on the how we stain using a light microscope. Because these are analogous to procedures used for light microscopy. For transmission electron microscopy, specimens must be cut very thin, and specimens are chemically fixed and stained with electron-dense material. So the staining agents here are with electron-dense material. So we use shadowing or coating specimen with a thin film of a heavy metal. So the shadowing, this is to prepare for electron microscopy. And another one is we have freeze etching or freeze specimen, then fracture along lines of greatest weakness like the membranes. So as you can see here in this figure is just to show you how the different preparation done for electron microscopy preparation. So if this is the plasma membrane, you can see here the nucleus of the cell, and you have to, using the knife edge, you have to fracture. So these are the fractured faces, and you have to coat it with platinum and carbon to have its shadowing direction, no, with the carbon. So and then before that it is still under sublimation, and then before it is the coating is incorporated, now you have the platinum carbon mixture inside the membrane. So this is an example under the electron microscope with an Ebola. It's a virus, no? Virus cannot be seen in a naked eye, but with using this electron microscope, you have seen here the image of what Ebola virus is. Another one, this is under electron microscope, is the fly head, no? The head of the fly. So it's very detailed and very prominent, no? So the scanning electron microscope is another type of microscope. It uses electrons reflected from the surface of a specimen to create image. So kanina the electrons are scattered, this one are reflected, and it produces a three-dimensional image of specimen surface features. So this is how is the different parts are illustrated. So from electron gun, you have there the condensing and the condenser lenses and the scanning oil, and the primary electrons are reflected here. And this is the specimen holder with the specimen. So in this part here, you can see the detector, the photomultiplier. This is under the vacuum system, and the scanning circuit, which contains cathode ray tube for viewing and cathode ray tube for photography. So there are also other newer techniques in microscopy. We have the confocal microscopy and scanning probe microscopy. So they have extremely high resolution and can be used to observe individual atoms already. So previously I have discussed to you here that under the light microscope, until mycoplasmas can only be seen. But with using electron microscope, they can already identify atoms, no? So in confocal microscopy, confocal scanning laser microscope is used. There's a laser beam used to eliminate spots on the specimen, and computer compiles images created from each point to generate a three-dimensional image. So this is just how the conventional light microscope versus the confocal scanning laser microscope is illustrated, no? With here, as you can see, you just take note the difference, not the depth of light collection, and this versus the for the biofilm surface. But in terms of um, the same, no, as you can see, the same focus of the image and the surface, but the view that you can see here is different from A to B, no? So it's more detailed here, and it's more clearer and nearer in the confocal scanning laser microscope. So this is another figure to show you the how is the laser light is embedded into the mirror and the scanner with the objective and with a cell here. So this is how the lenses are with a detector is illustrated. So this another one is the scanning probe microscopy. In the scanning probe microscopy, these are scanning tunneling microscope. These are steady current or tunneling current maintained between microscope probe and specimen. So there's a tunneling current or steady current. It's an up and down movement of probe as it maintains current is detected and used to create image of surface of specimen. So as differentiated from the rest, so it's just used as scanning tunneling. And for the scanning probe microscopy, you use atomic force microscope. These are sharp probe moves over surface of specimen at a constant distance. Up and down movement of probe as it maintains constant distance is detected and used to create an image. So this is just for this one is a sharp you use sharp probe moves, no? So again, the other techniques, no, the higher, the newer techniques for electron microscopy. This includes the scanning probe microscopy, which use the atomic force or sharp probe, and the scanning tunneling microscope, which uses the steady current or tunneling current. This is under scanning probe microscope, could be scanning tunneling or atomic force. And confocal microscopy, which uses confocal scanning laser. So these are just newer techniques in microscopy. In there are a lot, and there are even a lot now in commercial industry. Not only the scanning electron microscope and the transmission electron microscope. These are all under electron microscopy. So thank you very much for listening on this video. Please raise your questions by commenting or posting your questions on the Google Classroom. So for the next topic, we're going to have another separate video on which is all about metric system. So see you on the next video. Bye.